Totes at around 98, 99%. But it doesn't quite make it all the way to 100. And we also see that at the tissue level, when we're looking at an oxygen partial pressure of around 40 millimeters of mercury, right? We're thinking about a tissue that isn't working very well or anything like that. We're thinking about a tissue that's been using oxygen and has been working. Sorry about that. We see that it's still, we're looking at about 75% saturation of hemoglobin with oxygen. So that term deoxygenated, when we talk about blood that's returning to the heart from the tissues, is really not an accurate term because of what we have here. So this is kind of where we were at. We had ended by talking about affinity of hemoglobin for oxygen. How tightly do these bind to each other? And you guys had, I think we're pretty split, and I asked you to kind of give me your opinion. You guys were pretty split. So you said, yeah, I really want a hemoglobin with high affinity. I want to be able to grab as much oxygen at the alveoli as I can. And others of you had said, well, I'd really rather have my oxygen be able to get rid of, or my hemoglobin get rid of oxygen at the level of the tissue. So I want a hemoglobin with low affinity. And we saw that there's this sort of trade-off, right? We can't have both. We can't have one that grips oxygen really, really tightly at the level of the alveoli and one that dumps the oxygen really easily at the level of the tissue. We have to pick one. But there are some factors that can change the affinity of hemoglobin for oxygen. And that's kind of where we left things last time, right? We said that there are some factors that can shift the curve to the right, okay? So if we had a curve that had been shifted to the right, what would be happening here is we would be doing what? Increasing or decreasing hemoglobin's affinity for oxygen. Be doing what? Reducing, decreasing, right? Hemoglobin's affinity for oxygen. So if the curve shifts to the right, we said that curve has a lower affinity of hemoglobin for oxygen. And how did we prove that to ourselves? What measurement did we take, or what number did we use to sort of describe that? Remember? I drew my arrow again right where I need it to be. You guys remember the measurement that we took to sort of confirm that? Remember that we measured something called the P50? P50 is the partial pressure of oxygen. Okay, the PO2 at which hemoglobin is 50% saturated with oxygen. Right? It's the P50 value, right? So if I go over on my y-axis here to 50% saturation, all right? If I come over here and I draw this little dotted line all the way across to where I intersect both of my curves, and then I go down, okay? I see that with the blue curve, the one on the left, I see that the P50 value is lower than it is for the green curve, the one on the right. What that tells me is that I'm able to get to 50% saturation faster and with less available oxygen, meaning that this curve has a higher affinity for oxygen. It's binding to it more tightly and more easily, whereas this one is a little bit more resistant to bind oxygen because it has a lower affinity, and as a result, we see that the curve shifts to the right, okay? So you're right, a right-shifted curve has a lower affinity for oxygen. This is kind of where we left things last week. Does anybody have any questions about what we talked about last week? Me either. Okay. Me either. All right. So let's go ahead then and let's take a look at some of those factors that affect hemoglobin's affinity for oxygen. Okay. The first one, and probably one of the more important ones, is pH. Okay, the pH of our blood is going to affect hemoglobin's affinity for oxygen. So the pH of our blood is going to affect hemoglobin's affinity for oxygen. Let me do this really quickly here. So factors. Factors affecting hemoglobin's affinity for oxygen. The first one is pH. Okay, so what we've done here is we've shifted the curve to the right. Remember we said that normal blood pH is around pH 7.4. So let's go ahead and use that blue curve as our normal curve right here. Okay, so this is going to be pH 7.4. Okay. What happens to the pH of our blood when we start doing something like exercising? When you start exercising, not only are you breathing harder, you're making more CO2. We'll talk about that a little bit more today. But you're also producing things like lactic acid, which are going to be transported in your blood. So your blood becomes more acidic under certain circumstances, and exercise is a really good example of that. When your blood becomes more acidic, does the pH go up or does it go down? It goes down, right? A lower pH means that something is more acidic, right? So let's go ahead and compare that to this curve right here. Let's just say that this is pH 7.2, okay? pH 7.2. So we see that when pH drops, when blood becomes more acidic, what we do is we have a shift to the right, okay? So more acidic means lower affinity. Okay. More acidic means lower affinity, right? Does that benefit us during exercise? When might you want more oxygen getting to your tissues? So a lower affinity, we said last time, is going to be easier to deliver oxygen to our tissues, right? So when might you want more oxygen getting to your tissues? During exercise, yeah. Isn't that cool, right? How that works, right? During exercise, when the pH of our blood drops, the affinity of hemoglobin for oxygen lowers just a little bit, and it becomes easier to deliver oxygen to our tissues, okay? This particular shift that we see right here is called a Bohr shift in the curve, okay? This is called a Bohr shift. If you've taken chemistry and you've heard that name Bohr before, you heard it in relationship to the physicist Niels Bohr, who played a really instrumental role in describing the structure of atoms, nuclei and electron clouds and stuff like that. Turns out that Niels Bohr also dabbled in hemoglobin physiology, so this is named after the physicist Niels Bohr, just as you learned about him learning about the structure of atoms. Turns out he was a busy guy. Okay, let's look at a couple of other examples here. Any questions about that? Okay. Incidentally, a rise in pH would shift the curve to the left as well, but a drop in pH is significantly more likely than what we see than a rise in pH. So we're just going to kind of put physiologically realistic examples on the board. Okay. Another example, or another thing that affects the affinity of hemoglobin for oxygen is temperature. Okay. When we change the temperature of our blood, it's going to affect the affinity of hemoglobin for oxygen. It doesn't change a whole lot. We are what we call homeotherms, and we maintain a relatively constant body temperature, but it can change a little bit, right? So let's imagine a scenario, not necessarily, this might be a little bit out of the realm of physiological possibilities, but let's imagine a scenario where this is normal body temperature of 37 degrees Celsius, which is your normal body temperature. And then let's go ahead and raise the temperature just a little bit up to like maybe 43 degrees Celsius. Okay. We would again see a shift to the right, okay? A lower affinity of hemoglobin for oxygen. Okay. So warmer temps, warmer means lower affinity of hemoglobin for oxygen. Okay. This is probably a little bit of an extreme example. If your body temperature gets up to 43 degrees Celsius, you really should get to the doctor if you're still conscious. So that's a very high body temperature, but small changes still can affect the affinity, right? So when's an example of a time when your body temperature might increase? What? Fever? So, yeah, for sure, right? Fever. That makes sense, right? You probably want more oxygen getting to your tissues during that time, because if you have a fever, it means that your cells are, your immune system is fighting a foreign invader, right? And they're going to need energy and oxygen to be able to do that. What's another time when your body temperature might rise? Exercise. Oh, look at that, exercise again, right? Almost as if we're seeing a little bit of a trend right here, right? Yeah, exercise is another example of when we see this sort of thing occurring, right? So we see that temperature also can lower that affinity, then deliver our more oxygen to our tissues during a time when they need it the most. And then finally, the last one that I want to talk about is partial pressure of CO2, okay? Partial pressure of CO2. So CO2 building up in your blood, okay, can also reduce the affinity. So normal PCO2 is around 40 millimeters of mercury, okay? So let's just go ahead and put that up on here. Normal PCO2 normally is around 40 millimeters of mercury, okay? But when that CO2 level rises, let's go ahead and put a number on this, like 80 millimeters of mercury. When PCO2 rises, we see that we have a decrease in affinity, right? So rising a higher PCO2 means a lower affinity of hemoglobin for oxygen. What's a time when your CO2 levels in your blood might rise?
Exercise.
Oh, there you go, right? Exercise, very good, right? Yeah, right? If your tissues are working harder, they're making more CO2, which is a metabolic waste product of aerobic respiration, and they see that it rises, right? So during exercise and other times, but during exercise especially, we see this shift, this decrease in the affinity of hemoglobin for oxygen, and that favors the release of oxygen at our tissues from hemoglobin, right? Keep in mind that because of the asymptote up here, even though we're shifting the curve to the right, we're making it easier to deliver oxygen to our tissues, we're not really affecting the maximal saturation, right? We're keeping that pretty high. Thank goodness for the asymptote in this curve. Okay. So 3 really important factors that affect the affinity of hemoglobin for oxygen. Are there any questions about that? Okay. I want to talk about one more thing as it relates to this curve, and then I promise that I'll leave it behind. I probably have given way too much time to it as it is. But I want to talk about one more thing that I think is really important. You guys will remember that we had drawn the structure of hemoglobin on the board last week. I'll put it way down here so that it isn't in our way. But you guys have remembered that we have 4 subunits of hemoglobin, right? So we have our 4 heme groups, each with an iron in it. So Phe, Phi, Pho, Fun. Hemoglobin has 4 subunits in it. Right, we had our 4 subunits. And I had written on the board last week, do you remember what the names of these subunits are? What was that? When it is bound to when it's bound to oxygen, we do call it oxyhemoglobin. But these 2 subunits have names.
Alpha, beta.
Alpha and beta, right? So I've written on the board that adult hemoglobin, right? We said that adult hemoglobin was made up of 2 alpha subunits and 2 beta subunits. Okay? 2 alpha subunits and 2 beta subunits, right? So those were our subunits there, and I did differentiate it as adult hemoglobin because throughout our development we make different types of hemoglobin. We have genes in our DNA that code for different subunits. And we make very briefly, early on in our development, we make a type of hemoglobin called an embryonic hemoglobin. We don't make it for very long at all. Throughout most of our in utero development, though, we make a type of hemoglobin called a fetal hemoglobin. So fetal hemoglobin, okay, is made up of 2 alpha subunits. Those stay the same. Okay? Those stay the same. Those 2 alpha subunits stay the same. But the beta subunits are replaced with gamma subunits. And gamma kind of looks like the letter Y. They're replaced with these gamma subunits. Okay? The function of them on the surface really is no different. Those gamma subunits still bind to oxygen just like the beta subunits do. So from a functional point of view, fetal hemoglobin, just like adult hemoglobin, still binds 1, 2, 3, 4 oxygen molecules. But what is different is the affinity that those gamma subunits have for oxygen, right? So the affinity of those gamma subunits for oxygen is such that if we were to compare, one more comparison, if we were to compare a maternal curve, right, an adult mammalian curve, this is a maternal curve, to that of her fetus, we would see that the fetal curve shifts to the left. Almost got myself there. The fetal curve shifts to the left. So let's go ahead and put one more curve here on the board. We can see that the fetal curve shifts to the left. There we go. Almost got myself there. I'm a bit distracted, apparently. So we have a situation that looks like that, where a fetal curve actually shifts itself to the left, having a stronger affinity for oxygen. Okay? We had talked a little bit in the circulatory section about the fetal blood pattern, right, and the way that the mammalian fetus receives blood from mom. Okay, remember it's getting its oxygen, not blood, oxygen. It's getting its oxygen at that placental sort of interface right there, and the PO2 down there is really, really low, right? We likened the amount of oxygen in the at the placenta to essentially what we would encounter on the summit of Mount Everest, right? So what this fetal blood is doing, what this fetal hemoglobin is doing, is it's trying to grab as much of that oxygen as it can with this really high affinity hemoglobin. Is there a trade-off because it can't release it as easily to its tissues? Sure, right? But the PO2 is so low, right? The PO2 is so low that it ends up benefiting the fetus. There are some mammalian species, humans are not one of them, where we will actually see that mom will make extra metabolites during her pregnancy, where it will actually shift the mom's curve over to the right even more, so that mom is trying to dump as much oxygen as she can and baby's grabbing as much as it can, because the difference between these two curves is only going to give more oxygen to the baby. Humans don't do that as far as I'm aware, but there are some mammalian species like horses are a really good example, where mom will make all of these metabolites during pregnancy that will actually shift her curve over to the right even more, making it easier to dump oxygen to the baby. And then the fetus, of course, has this left shifted curve. So it's kind of an interesting one. Remember that the lifespan of individual red blood cell is only about an individual red blood cell is only about 3 to 4 months. Okay, so within about 3 to 4 months after being born, baby has replaced almost all of its fetal hemoglobin with adult hemoglobin, those 2 alpha and 2 beta cytokines. Huh?
How many months did you say?
About 3 to 4. I mean, the lifespan of an individual red blood cell is about 3 to 4 months. So if we assume that baby's born with all of this fetal hemoglobin, it starts replacing those immediately, about 3 to 4 months, and they probably are gone, by that point. Okay. Any questions about oxygen transport in the blood? Okay. Without hemoglobin, we're not gonna get enough oxygen to our tissues. Okay. With that said, then let's go ahead and talk a little bit about CO2 transport in the blood. We haven't talked about CO2 yet. I don't have as much to say about it, I promise. Even though CO2 is ostensibly way more important than oxygen in a lot of ways. So CO2 transport. Let's talk about CO2. Okay. Carbon dioxide, as you guys know, and as we mentioned a couple of minutes ago, is produced in very large amounts in our body, and also continuously as well, right? It is the waste product of aerobic respiration. Okay? So every single time you make ATP aerobically, which is how you make the vast majority of your ATP, you're making CO2 as a waste product. So the production of CO2 in your body literally never ceases. It's being made on a continual basis. All right? So we have to figure out something to do with all of this waste, right? We don't often, I think about CO2 as a metabolic waste product, but that's exactly what it is. Okay? So we have to figure out something to do with it. Turns out that of all of the CO2 that we make, we do transport some of it dissolved in our blood plasma. We talked a little bit last time about the solubility of a gas in the blood. Okay? It turns out that CO2 is a little bit more soluble in solution than oxygen is. So about 7% of the CO2 that we produce is going to be dissolved in our blood plasma. Have a lot to account for still, but about 7% of it is dissolved in our blood plasma. We haven't talked about this yet, and we're not going to do more than just mention it, but your hemoglobin also has binding sites on it for carbon dioxide as well. You can actually bind carbon dioxide to hemoglobin. And about 23% of the CO2 that we make is carried out of our body via hemoglobin. Okay. About 23% binds to hemoglobin. When we have hemoglobin plus CO2, we get something called carboxy. The rest of it, though, we have to account for, right? If you're doing the math with me here, 23 and 7 is only 30%. So what about the other 30%, or what about the other 70%? Okay, the vast majority of CO2. Okay, the vast majority of CO2 is going to get converted to carbonic acid. Okay, so the vast majority of CO2 is converted to carbonic acid. The vast majority is converted to carbonic acid. So in your guys' red blood cells, there is an enzyme called carbonic anhydrase. All right, so let's just imagine this scenario here really quickly. Okay, you've got a tissue right here, right? Your tissue is kind of right here. And we're producing CO2 in that tissue, okay? And that CO2 is going to diffuse into a blood capillary, right? There's your capillary kind of right here like this. And inside of that, you've got red blood cells, right? So we've got red blood cells kind of floating around in here. That CO2 is going to diffuse from an area of high concentration to an area of low concentration into your blood. Okay. At that point, about 7% of the CO2 is going to dissolve in your blood plasma. About 23% of it is going to diffuse into the red blood cell, and it's going to bind to hemoglobin. Okay. The other 70% is still going to diffuse into our red blood cells, but it's going to get converted to carbonic acid. So let's go ahead and take a look at that really quick. So we're just going to take our capillary and really blow it up here. Okay, so there's our red blood cell right there. So we're going to CO2 diffuses in here, okay? And then diffuses into our red blood cell. Okay? CO2 is going to combine with water, so CO2 plus H2O, okay, is going to be converted to carbonic acid, H2CO. Okay? This is carbonic acid. The enzyme that does that, the enzyme responsible for that, is an enzyme called carbonic anhydrase. The enzyme responsible for that reaction, which is reversible, is called carbonic anhydrase. I don't normally bring up enzyme names a whole lot, but a carbonic anhydrase is worth mentioning. Not only because of the massive amount of work that it does, right? Again, we're producing massive amounts of CO2, so this enzyme is working around the clock. Also for the speed that this enzyme works at, okay? To give you an idea of just how fast this enzyme has the ability to work, this reaction, converting CO2 and water to carbonic acid, carbonic anhydrase has the ability to do this chemical reaction 1,000,000 times per second. Okay? I mean, try just for a second to wrap your mind around that, right? 1,000,000 times per second is how fast carbonic anhydrase can work. Okay? Probably doesn't really work that fast inside of our bodies, but if we were to push it to its limit, its theoretical limit, it could work about 1,000,000 times per second. That's an astounding number. Okay, that's a truly astounding number with how much, and it's also a testament to what this enzyme has evolved to be able to do, right? It's it's a testament to how much CO2 that we're producing. In the red blood cell, that carbon dioxide is converted to carbonic acid, and then that carbonic acid diffuses back into the blood plasma. You've got chemistry. Can you tell me what acids tend to do in solution? In solution, in the blood plasma, H2 CO3 is going to do what? What acids tend to do in solution? Expand on that. Guess. Do they stay together? They don't, right? They dissociate in solution, right? Yeah, they dissociate in solution. So when this dissociates in solution, it becomes hydrogen ions plus something that we call a bicarbonate ion, which is HCO3 with a negative charge, right? This is a bicarbonate ion. Okay, becomes a bicarbonate ion. Hydrogen ions plus bicarbonate ions is what it dissociates into. When you guys have the buildup of hydrogen ions in a solution, okay, when we have the buildup of those hydrogen ions in solution, what does that do to the pH of a solution? pH is a measure of hydrogen ion concentration, right, in solution. And an increase in hydrogen ion concentration causes the solution to become more acidic, right? It causes a drop in pH, right? Okay. Hydrogen ions build up in solution, we have a drop in pH. pH becomes more acidic. Okay. So, as carbon dioxide diffuses into your blood, okay, the majority of it is converted to carbonic acid in your red blood cells because of this enzyme carbonic anhydrase. It diffuses back into your blood plasma where it dissociates into hydrogen ions and bicarbonate ions. But that buildup of hydrogen ions causes your blood to become more acidic. Okay. Now, with your lungs, okay, all of this reverses itself. Okay. The hydrogen ion and the bicarbonate ion are going to be converted back into carbonic acid, and carbonic anhydrase reconverts that carbonic acid back into water and CO2, and you exhale water and CO2. Okay? Mostly, but the water is not coming from the blood, it's coming from evaporation. But the CO2 is being exhaled this way, right? So at your lungs, all of this completely reverses itself. That's why I've drawn bi-directional arrows on all of these reactions, because they are all completely reversed, right? So at the level of our tissues, we see, let's just go ahead and rewrite that equation down here for everybody at this end of the room. Let's go ahead and so here at our tissues, okay, we see that CO2 plus water are getting converted to H2CO3 carbonic acid, which then dissociates into hydrogen ions plus bicarbonate ions, okay? And then in our lungs, okay, our tissues, it's moving in this direction, okay? And then our lungs, we see that it's moving back in that direction, okay? Does that help? Let me make these arrows red. Right, in our lungs, it's moving in that direction. Okay, so in our tissues, we're receiving that carbon dioxide, we're converting it, the pH of our blood is dropping, and then at the level of our lungs, we see that the reaction reverses itself, and we actually are exhaling that CO2. Okay. So what this means is that what we have to do is we generally have to match the rate at which we're breathing to the rate at which we're making CO2, right? If I want the pH of my blood to stay level or even or the same, right? If I want the pH of my blood to remain at homeostasis, my rate of respiration, the rate at which I'm getting rid of CO2, has to roughly match the rate at which I'm making CO2, Does that make sense? So what happens if I do something like this? Right. The pH of my blood is dropping when I hypoventilate. Okay? The pH of my blood drops when I hypoventilate. So hypoventilation, okay? Hypoventilation is going to lead to a state that we call respiratory acidosis. Hypoventilation leads to something that we call respiratory acidosis. Okay, now let's do the opposite. I can't do that for anywhere nearly as long. That one really hurts. Hyperventilation. Now, what I'm doing is I'm shifting the equation in this direction, and I'm actually breathing off CO2 faster than I'm making it. So what is the pH of my blood going to do? It's going to become more basic. It's going to come up, right? So hyperventilation leads to a state called respiratory alkalosis, because the pH of my blood becomes more alkaline when I hyperventilate. Okay? I don't do it so much anymore, but you know, it used to be you would go to the emergency room because you're hyperventilating. You know, you're having a panic attack or something like that. It's not out of the realm of possibilities. But you go to the emergency room, you're hyperventilating, right? What are they going to do to at least initially help to treat you just a little bit? They're going to give you a paper bag to breathe into. What are you doing when you do that? If you're hyperventilating, what you're doing is you're blowing off excess CO2 and you're in a state of alkalosis. When you breathe into a paper bag, what about the gas that you're breathing? Every time you exhale into a paper bag and inhale again, you're breathing CO2. Or you're breathing CO2. And what's that going to do to the pH of your blood? It's going to bring it back down to normal levels, right? So the very first order of treatment is to correct the pH of your blood. Okay? So that's why you breathe into a paper bag. If you go to the emergency room for one of those, it's going to be the most expensive paper bag that you ever buy in your lifetime. But it's a very fast way for us to correct and change the pH of your blood. It only takes a few seconds for that pH of your blood to change. So when we think about control of respiration, right, we know that the rate and depth of our breath changes on an almost continual basis, right? I know that we talked about respiratory volumes and capacities last week, but even just sitting here, you're probably changing the rate and depth of your breath, as you're sitting here. Probably so much so that you aren't even really noticing it, right? But you probably are. And the reason behind that is because you're making these minute changes to the rate and depth of your breath so that you can control this chemical equation and keep the pH of your blood at a more or less constant state, right? In fact, the most important variable when it comes to the rate and depth of your breath is the pH of your blood plasma, right? You might think to yourself, right, it's oxygen, right? Dr. Russell was holding his breath a couple of minutes ago. He finally had to stop holding his breath. We all know that that's the case, right? You can only hold your breath for so long before you have to take a breath, right? How many of you think that that's oxygen forcing you to do Before today, how many of you thought it was oxygen? Right, it's not, right? Oxygen levels don't drop as fast as we might think, right? That hemoglobin-oxygen dissociation curve keeps quite a bit of oxygen in your blood, right? But it's the pH of your blood because of CO2 buildup that is what's changing, right? So pulse oximeters, those little things that we put on our finger to look at oxygen concentration, if we had one of those that could measure CO2 in our blood, we would see that that would be what's changing, right? If we can measure pH in our blood, that would be what's changing. And that's why your brain eventually tells you, oh no, you've got to take a breath, right? The pH has gotten too far out of homeostasis. We have to correct that. Okay? Why is pH so important? What does having our pH out of homeostasis do? How does it form? It forms proteins, right? Proteins, every protein in your body, and proteins literally do everything in your body, right? They have a very specific erase my hemoglobin, but all of the proteins in our blood or in our body have very specific three-dimensional shapes. If that three-dimensional shape is off just a little bit, what's going to happen is that protein's not going to be able to do its job. One of the things that affects the shape of the protein is pH, and if the pH is out of homeostasis, it can cause our proteins to do what we call denature or fall apart, and that can be awful for us. So we have to keep pH in our normal range of function. Okay. So the single most important factor that regulates the rate and depth of our breath, the single most important factor that regulates the rate and depth of our breath is the pH of our blood. Okay? The single most important factor that regulates the rate and depth of our breath is the pH of our blood. Okay? Oxygen has to get a heck of a lot lower than you would think that it does before it makes you start breathing faster. Okay? Oxygen can go really low before it affects the rate of our breath. CO2, and as a result, the pH of our blood is way faster. Question? Sorry? First part of what? Sorry. The single most important factor that regulates the rate and depth of our breath is pH. Okay? The single most important factor that regulates the rate and depth of our breath is pH. Oxygen, yeah, it does play a role, but like I said, oxygen has to get way lower than you would expect it to be before it actually causes us to breathe faster. CO2 is going to be way faster to accumulate, and the pH of our blood is going to change way faster. Okay. So I know we aren't spending as much time on this, but this is arguably way more important than oxygen transport in our blood, right? Because oxygen, we can deal with low oxygen. This is just like instantaneous, right? Holding my breath for 30 seconds, I would actually measure a change in the pH of my blood, okay? Such a profoundly powerful and fast physiological response. It's really, really pretty crazy. All right? Okay, any questions? Yeah.
Just to clarify, you said hyperventilation is when you go from the lungs.
So when you hyperventilate, yeah. So sorry to interrupt, but yeah, when you hyperventilate, what you're doing is you're essentially getting rid of CO2 faster than you're making it. Okay. So what you do is you kind of shift the imagine if this helps. Imagine that this whole equation is sitting on like a seesaw, right? So this whole equation is sitting on a seesaw, like this. And you've got your little balancing point right here, right? So what happens is when you blow off CO2 faster than you're making it, what happens is that the seesaw kind of shifts in this direction, right? And imagine, because the hydrogen ions are there, what happens is we're getting more alkaline, right? We're getting more basic, right? When you hold your breath and you allow CO2 to build up, what it does is it shifts the seesaw or the teeter-totter in this direction, and the pH of your blood goes down, right? So if the seesaw shifts in this direction, we become more alkaline. If this isn't helping you, ignore it. If the seesaw shifts in this direction, it becomes more acidic. Right? So imagine that it's just sitting on a seesaw, and if we're getting rid of this faster than we're making it, whoop, it shifts like that. The blood becomes more alkaline. If we are holding our breath and we allow this to accumulate, right? What happens is the seesaw does this, right? Goes whoop like that, and our blood becomes more acidic. Does that help? Okay. Okay. What do we call these in Southern California? Are they seesaws or teeter-totters in Southern California? Teeter-totters? Okay. Okay. That's what I grew up calling them. But, you know, there's regional terms for things. So, seesaw, teeter-totter. Is there another name I'm not aware of for them? No? Not that I'm aware of. All right. All right, you guys. Let me think here just for a minute. Okay. Okay, so real quick, and then I want to put a bow on the respiratory system so that we can start to talk about the renal system today. Real quick, I want to very quickly mention the control of respiration, right? So breathing is kind of an odd thing if you think about it, right? It's controlled, your act of breathing, right, inspiration and expiration, is controlled entirely by skeletal muscles, which at this point we know are voluntary, right? So that's why I can opt to hold my breath. That's why I can opt to hyperventilate if I want to. But at the same time, breathing is also something that you don't really have to think about doing, right? You just do it, right? So it's a little bit of a weird thing in a lot of ways because you don't really have to think about it. In your pons of your brain, okay, in the pons you have these neurons called central pattern generators. So we have let's see, control of breathing, right? Of breathing. In the pons, you have neurons called central pattern generators. We're going to simplify this tremendously. There's a lot to actually say about this, but we're going to be out of time if we don't simplify things a little bit. In your pons, there's these groups of neurons called central pattern generators. And the best way to sort of think about these central pattern generators is to sort of think about them like the pacemaker cells in the heart, right? They launch action potentials at very regular intervals that triggers your diaphragm to contract, your intercostal muscles to contract, and you take a breath, right? But because these are skeletal muscles, it means that your cerebral cortex can take over, right? And that's why you can do things like hold your breath, or you can do things like hyperventilate. Okay? But at the end of the day, these central pattern generators are what set your rate of respiration. These are heavily influenced by things like pH of your blood, okay? They're heavily influenced by things like oxygen concentration. Again, oxygen can get really low before it does anything to these central pattern generators, but at the end of the day, these are what have the major control over your rate and your depth of breath. Okay, and eventually they can actually override what your cerebral cortex is saying, right? Which is why when you're holding your breath, you might say, I want to keep holding it, I want to keep holding it, I want to keep holding it. But eventually you're like, oh, I gotta take a breath, right? Because these central pattern generators eventually will override signals from the cerebral cortex and you'll be forced to take a breath, right? That's why little kids, little threats of holding their breath until they pass out when they don't get their way never work, right? If a kid ever does that, just say, go for it. You know, I know that your central pattern generator is going to take over. Just make sure they don't hit their head on their way down. A strong-willed kid might actually make themselves black out for a second. But don't worry, they'll start breathing immediately. Because of central pattern generators. Yeah? No, I mean, I mean, some babies will, but these eventually will, you know, take over. So, I mean, yeah. Okay? Yeah.
So like for individuals who are, let's say like swimmers who are training to hold their breath,
Mm-hmm.
is this central pattern generator something that can be like elastic, like you can train that over time?
Yeah, so there is a little bit of elasticity to it, right? So we can manipulate it a little bit. We can learn to shut it off for a little bit longer, like you said, for long breath holds and stuff like that. Also, remember, it's the pH of your blood that affects the rate, the length that you can hold your breath for. So you can manipulate that, right? Hyperventilating before you hold your breath, driving the pH of your blood up, will take it longer to become acidic to that critical level, right? I had a group of students once who did that as an experiment. I used to make my students actually conduct experiments for their final projects and report the results. You guys got lucky. But they actually did this for their final project. They would have they had like 40 people, classmates and family members and stuff like that, hyperventilate they had them hold their breath. They said, hold your breath for as long as you can. And then they had them hyperventilate for 30 seconds and then hold their breath for as long as they could. And turns out that they could hold their breath for like 30 or 40% longer after they hyperventilated, right? So you can actually manipulate the pH of your blood to hold your breath for longer and things like that. But I think it is a little bit on the plastic side as well. Like we can actually reset what the critical levels are for some people. And there's probably like a genetic predisposition there as well. So some people are probably genetically predisposed to holding their breath longer than others. But I think that there is some plasticity there, yeah. Okay. There's a lot more that we could say about this. The medulla also plays a little bit of a role, mostly by smoothing out your rate of breath, but it's the pons where we find those central pattern generators. And interestingly enough, this is one of the areas of physiology that we still don't have completely figured out. We understand the basics of how it works, but man, this whole control of breathing and what goes on in those central pattern generators is still a pretty big black box of physiology. We still don't completely know how all of it works. So because we are limited on time and because we don't completely understand it, we're just going to kind of very generally introduce it and we're going to kind of leave it at that. Okay. Yeah.
Sleep apnea would stop breathing.
Uh-huh.
This would make you start breathing?
You know, yes, this is why you would start breathing again, because of pH changes in your blood, right? So this is where that comes into play, right? Exactly, yeah. Yeah.
So the pons, if they would recognize the pH levels, that's what makes you.
Yes. So the pons is what receives that message, and it's, you know, there's pH sensors, there's CO2 sensors, there's oxygen sensors. You know, I suppose it's conceivable that oxygen levels in the blood could drop really low, and CO2 and pH levels could still be normal. Those would be weird circumstances, though, but it is conceivable that that could happen. So, yeah. So we still have oxygen sensors in there as well, but mostly pH is the biggest and most important variable. Okay. Right. anything else? All right, well, I think that we've wrapped up respiratory sufficiently for me. We will start renal today in lab, and then we will go ahead and start to talk a little bit about it, and we'll kind of go from there. Renal's a little shorter than respiratory.